This video is going to look at the structure of proteins. Proteins are naturally occurring condensation polymers made up of amino acid monomers. Amino acids are molecules that contain an amine group and a carboxylic acid group. And the amino acids are joined by what the biologists call a peptide link. We've come across this before when we talked about polyamides. It's the amide bond in chemistry, and that's a carbonyl group joined to an NH group. So the main chain of the protein has carbon and nitrogen atoms in. Now you can break the main chain by hydrolyzing the peptide bonds, as we saw with polyamides, and that releases constituent amino acids. Proteins have complicated three-dimensional shapes, and their function, what they do in the body, whether it's catalyzing a reaction or acting as a receptor for a small molecule, or as an antibody against an invading virus or bacteria, those functions depend on their three-dimensional structure. So when we're describing the structure, we look at different levels of structure. The simplest is the primary structure. We then build up that primary structure into building blocks of the overall structure, which are called secondary structure. And then the overall three-dimensional shape of a polypeptide chain is the tertiary structure. Now, some complicated proteins have more than one polypeptide chain. So hemoglobin is an example, the protein that carries the oxygen in your blood. That has 4 polypeptide chains, and the way those 4 chains are arranged is called the quaternary structure. The primary structure is the sequence of amino acids in the protein, and that's determined by your DNA. The DNA tells the cell what order to put the amino acids in. Now, biologists use different codes for describing the primary structure. One of the codes is a 3-letter code. And each of the 20 naturally occurring amino acids in human proteins has a different 3-letter code. Here's an example of a short sequence of primary structure. So we have, starting from the left-hand end, aspartic acid, alanine, cysteine, alanine, phenylalanine, glycine, alanine, and glycine. Each amino acid is linked to the next by a peptide bond. The diagram shows 4 amino acids linked by peptide bonds. I've shown in red the side chains of the amino acids. Those are the parts of the structure that vary. So in glycine, if R1 was glycine, you'd have a hydrogen atom. If R2 was alanine, you'd have a methyl group. And if R3 was cysteine, you'd have a CH2SH group. All the bonds in this structure that I've drawn are covalent bonds. So the primary structure is relatively stable. It takes quite severe conditions to break the peptide bonds. So for example, if we want to hydrolyze a protein with acid, we have to reflux the protein with 6 mol per dm cubed hydrochloric acid. The next level of structure is the secondary structure. Now, if you like, this is the scaffold that the total three-dimensional shape of the protein is built on. So it's the framework for the protein. And what you find is that the secondary structural elements that are shown here are used in all proteins from all different organisms, from the simplest virus right through to humans. So evolution's come up with a regular structure that works, and it's used throughout organisms. So the two secondary structural motifs, types of secondary structure that you need to know about, are the alpha helix and the beta pleated sheet. In both the alpha helix and beta pleated sheets, the primary structure of the protein folds back on itself, and that it's held together by hydrogen bonds between the carbonyl in one part of the chain, the C double bond O group, and that hydrogen bonds with the NH of the amide peptide link in a different part of the primary structure. In the alpha helix, the primary sequence is coiled around an axis, like a coiled electrical cable on an old landline telephone or on a computer. There are 3.6 amino acids per turn of the alpha helix, and as I've mentioned, the structure is held together by hydrogen bonds between an NH group in one turn and a CO group, C double bond O, in the next turn along the helix. The alpha helix provides a stable structure on which to put the amino acid side chains, and they stick out from the alpha helix, away from the helix, so that they can fulfill different functions in the protein. In the beta pleated sheet, you have a flat polypeptide backbone, and that folds back on itself, and you have another strand of polypeptide running parallel to the first one, and you get hydrogen bonds between the CO, C double bond O group in one strand and the NH group in the other strand. And in the case of the beta pleated sheet, the amino acid side chains, which I showed in red in my structure on the previous page, stick up and down from the flat beta pleated sheet, so then they can make bonds and interact with other atoms in the protein or in other molecules. The key thing to remember about secondary structure is that it is held together by hydrogen bonds between the C double bond O and the NH of different peptide links. So the diagram here shows the hydrogen bonding that holds these groups together. Hydrogen bonds are much weaker than covalent bonds, so secondary structure can be broken up, disrupted by more gentle heating or changing the pH of the protein. So if you're doing biology, you'll know that enzymes work best at a certain temperature and at a certain pH. And if you go outside fairly limited ranges, they stop working, they denature. And that's because the structure of the protein is affected. remember when drawing the diagram of hydrogen bonding that you need to show the lone pairs, you need to show the dipoles, and you have to indicate the hydrogen bond. So the secondary structure and other parts of the protein can fold up to give you the overall three-dimensional shape of the protein, which, as I said, is essential for the protein's action. There's lots of techniques that have been used for studying the three-dimensional shapes of proteins, and the two most important are X-ray crystallography, where you grow a crystal of the protein and then fire a beam of X-rays at it and interpret the pattern that of spots that you get from the crystal. And then NMR spectroscopy, which is a technique that's done in solution. The tertiary structure is complicated and it's held together by lots of different types of bond and interaction. So this diagram summarises the types of bonds and forces that can hold proteins' three-dimensional shape together. So you have ionic bonds between charged parts of the structure. So here we've got an amino acid. On the left of the diagram you have an amino acid that has an amine in the side chain, and that's charged, and you've got a second amino acid that has a carboxylic acid side chain, and that's also charged, and the plus and minus charges can form an ionic bond. In the middle of the diagram there's a picture of two non-polar amino acid side chains, and they're forming van der Waals forces between the methyl groups. To the right, the sulfur-sulfur double bond, that's formed by two cysteine residues that have side chains that have reacted together. And then finally, on the right hand side of the diagram you have two side chains of two amino acid side chains that have formed a hydrogen bond. Van der Waals forces are formed between non-polar amino acid side chains, such as leucine, and hydrogen bonds can either be performed between the carbonyl and NH groups in the main chain of the protein in the backbone, or between amino acid side chains that contain groups like hydroxyl groups, which are found in serine, or the CONH2 group that's found in glutamine. The ionic bonds, as I've said, are formed between amino acid side chains that have charged groups in them. So things like NH3+, which will occur in lysine, and C double bond O minus, COO minus, which you find in aspartic and glutamic acid. So the charge state of these groups will be affected by the pH, as we've seen when we talked about the isoelectric point. Disulfide bond is formed when two cysteine residue side chains react together. So the cysteine, the SH is the sulfur equivalent of an OH group, hydroxyl group, and these can react together to form a disulfide bond. Now this is a covalent bond, so it makes a very strong link between different parts of the protein main chain. It helps to stabilise the tertiary structure. you get lots of disulfide bonds in proteins from organisms that live at high temperature. So the enzymes that are used in automatic washing powder, the lipases that dissolve fats and the proteases that dissolve protein, they're based on proteins that are found in thermophilic organisms, ones that live at high temperatures, and their proteins are stabilised by disulfide bonds. Generally, it's quite easy to disrupt the tertiary structure of proteins, either by heating them. So when you cook an egg, you change the structure of the protein in the egg, and that's irreversible. You can also change the structure by changing the pH or by changing the polarity of the substance. So if you add extra salt, make the polarity of the water higher, then it will affect the structure of the protein. So that completes a quick review of the structure of proteins, how they're organised in different levels, and in the next video we'll go on to see how that structure is used in a particular type of protein, the enzyme.